What is experimental evolution? By the end of this slide presentation, you should be able to explain how experimental evolutionary studies differ from traditional evolutionary studies, differentiate between the three population propagation models and the type of evolution they will exhibit, define allele replacement in the context of experimental evolution, and identify how the Malthusian parameter is used in experimental evolution. Experimental evolution is the study of evolution in controlled laboratory environments. It allows evolutionary dynamics to be monitored in real time. It differs from traditional evolutionary studies by testing evolutionary hypotheses prospectively rather than retrospectively. This means rather than analyzing molecular data first to see if it matches a hypothesis or prediction, a hypothesis is made and then the outcome of an experiment is analyzed to see if it aligns with the hypothesis. Experimental studies also differ from traditional molecular evolutionary studies as observers are able to see evolution in action and maintain ancestral populations as well as their descendants. Cryopreservation is one way in which ancestral populations are maintained. Most experimental molecular evolution studies use microorganisms for predictable reasons. They have short generation times, can be propagated under controlled conditions, can attain large population sizes, possess small genomes, can be cryopreserved, and easily fit in the laboratory. A few experimental molecular evolutionary studies have used animals, such as Drosophila flies and guppies. Simple experimental evolution experiments start with the founding population of organisms that can be genetically heterogeneous or consist of a single cloned organism. This population is then expanded to create replicated populations. These replicated populations are then propagated in an experimentally determined environment. Samples are cryopreserved from the founding population and all subsequent populations at designated time intervals to track genetic change. The figure on the right side of the slide visually represents this. Snowflakes are used to indicate cryopreservation steps, and different colors in the flasks are used to represent populations that have undergone genetic change from the founding population, which occurs red in color. From these cryopreserved samples, analyses are performed to track the temporal dynamics of genotypic and phenotypic changes. These can be used to determine the evolutionary paths of mutation accumulation. There are three main models for propagating populations in molecular evolutionary experiments. Model A is a mutation accumulation experiment. In this type of experiment, intentional population bottlenecks are created to maximize fixation of neutral mutations. This is accomplished through colony picking on agar plates. Model B uses continuous culture to maintain population size using a chemostat, where there is a constant flow of inflow of nutrients and outflow of cells. In Model C, serial transfers are made from portions of the population to fresh media once their nutrients are depleted. In order to prevent populations from decreasing or staying stable, however, transfers can be made before the nutrients are depleted to allow for constant population growth. In models B and C, adaptive evolution is possible because ample genetic diversity is maintained. Evolution in Model A will proceed through random genetic drift. In allele replacement experiments, alleles derived from evolved populations, here represented in red, can be transplanted into ancestral genomes, represented in blue, and ancestral alleles can be transplanted into evolved genomes. This allows researchers to examine these allelic changes independently of any other genomic changes that have taken place over the course of the experiment. The most important variable that can be determined from experimental evolution is mean fitness. Mean fitness is useful for determining if mutations made in the molecular evolutionary process have increased or decreased the overall fitness of the organism. In wild populations, it is very difficult to estimate mean fitness, but in the lab, it can be estimated using the Malthusian parameter, which represents the exponential growth of a population. It can be described by the equation dn over dt equals alpha n, where n is the population size, dn over dt is the increase in population size per unit time, and alpha is the Malthusian parameter. It has proxies such as the realized Malthusian parameter and the solution to the Malthusian parameter. This figure shows two different ways a strain could have a Malthusian parameter that is 10% lower than another. In example A, strain 1 grows faster than strain 2, and in example B, strain 1 has a shorter lag phase than strain 2. In both, strain 1 has a Malthusian parameter that is 10% higher. These type of comparisons are often made between founding populations and evolved populations in experimental molecular evolution.